hate when it's like that because then I can't get to my thing. I don't know why sometimes it does that. View. Slideshow. There we go. Alright, so my crappy grainy picture I took yesterday with my cell phone. Jeff Smith's going to take better pictures for me. So there's our glowing E. coli, right? And which we did achieve that, right? We did transform some bacteria. They are glowing, right? Is the plate that's supposed to glowing? Yes, right? So use your little pen light and you can look and you will see, right, some dots of glowing. So we expected, and why is that plate? None of the others are glowing, right? You can go ahead and check with your UV light, right? But as I have pictured here, this is the only one that's glowing. Why is that? It's the only one that contains the arabinose, right? Which binds to the repressor, allows the promoter to become available for RNA polymerase. They make the RNA, they make that protein, right? It's under that operon for arabinose, right? It's under that control for green fluorescent protein. So this plate, right? Both these plates, they were plated with transformants, right? They have the DNA, so they should be able to make green fluorescent protein. So plate C isn't making it because it doesn't have the sugar, right? The arabinose. But do you notice how it's just little dots, right? On this plate that are glowing? Not all the growth on that plate is glowing, correct? Right? So look at that plate again more closely with that. And you'll see that there's growth all over that plate, right? But only some colonies are actually glowing. So the ones that are glowing we know were transformed, right? What about all that other bacteria on that plate? They're not transformed, are they? Because if they were, what would they be doing? They'd be glowing. So there's another clue on one of our other plates that lets us know what went wrong with this experiment. What other plate does not have expected results on it? Actually, two of them, but one of them that really stands out. Right? All the rest of the plates pretty much look the same, right? Are all of these plates supposed to look the same? No. Which one clearly, drastically, does not at all look like as expected? B. Okay? So B, what was it supposed to look like? There wasn't supposed to be any growth at all, right? Because did these bacteria have the DNA? No. What are these plates supposed to contain? What's B supposed to have? Ampicillin in this plate, right? And these bacteria do not have, right? They're DNA negative, right? They shouldn't have the BLA gene. They shouldn't be making beta-lactamase. They should not be able to destroy ampicillin, right? Well, if they don't have that ability, how is it they're growing on this plate? What do you think? What could have went wrong here? Contamination with something that can grow in the presence of ampicillin? That would have to be that contaminant. What's the chances of that? That's a broad-spectrum antibiotic. Kills both gram-positives and gram-negatives. We used the wrong plate. You're getting warmer. The ampicillin in it was bad. Yes. 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 Well, not really necessarily expired. We had some issues with electricity, and my stock got thawed out and refrozen, and too many times, so it didn't work. So I got some fresh stock from another lab, and I actually made up some broth, right? And what I did was, and I hadn't taken pictures of those yet, I'm going to have Jeff help me with that, because Again, my cell phone doesn't do as good as a real camera, especially with stuff like this. But what I did is I scraped a little bit off a set of plates from lab, and I put them in broth containing ampicillin. And then I came back the next day and I plated them onto plates. And the ampicillin in that broth did kill the DNA negative bacteria, the non-transformed bacteria, and nothing grew on that plate. So I have those in the incubator. I'll take them out for you guys in a moment. And for these plates, now what you don't notice here, in here is, right, remember these should only be transformants. And we really can tell the transformants on this plate because they're the only ones that are glowing. But on this plate, because there's no arabinose, we can't see the ones that were transformed because they're not producing the green fluorescent protein. So when we shine the UV light on them, we can't see the transformants, right? They're there, but they're probably also getting outcompeted by the non-transformants, which was the case here, right? Most of this plate is covered with non-transformants. And then you have the little colonies that are glowing. So had we done transformation in the presence of ampicillin, on both of these plates, we should have just seen colonies. We shouldn't have had a complete lawn of growth, right? Because the efficiency of this is about 75 colonies, right, per transformation. So on each one of these plates, we would expect about 75 of these little dots. So when you look at this one came out pretty good, right? It was pretty efficient. I mean, there's probably more than 75 little dots there, right? I got really nervous, though, because when I looked at the plates a day or two later, because there were so many non-transformants, the transformants, it took them a while to grow up and start glowing. So I thought maybe our transformation didn't work at all. And I was a little freaked out. But the good news is it did work. The bad news is that ampicillin was bad, right? And so it didn't stop the growth like it should have here on our B plate. And it did not kill off the non-transformants on these plates so that we were selecting for just transformants. Does that make sense to you guys? Okay. So last lab today, right? Yay! This is it, right? So yeah, this is what we... Okay, so let me go ahead.